So the muscular system is chapter 9. The muscular system, we're mostly going to be concerned with the skeletal muscle, okay? Because skeletal muscle tissue is what makes up the skeletal muscles, which are the organs of the muscular system. The skeletal muscle, you probably remember from our histology section, is the muscle that is attached to the bones. That's why it has the skeletal part to it. It's the muscle that's attached to the bones. And this is voluntary. What do we mean by voluntary as opposed to involuntary? You can control it, right? So you can think about moving your arm and move your arm, right? But when we look at cardiac muscle and smooth muscle, we know that we can't control these muscles consciously, right? You can't just think about it and make your heart stop beating. You can't just think about it and make your stomach stop churning and moving your food around, right? So these are involuntary muscles and skeletal muscle is voluntary. Okay, we talked about cardiac muscle previously, right? You looked at the tissue slide of cardiac muscle. It's found only in the heart, it's involuntary, and this is what pumps the heart. We will come back to cardiac muscle when we get to the cardiovascular system, okay? So the cardiovascular system will study the heart and the blood vessels. Smooth muscle. So smooth muscle is found in the walls of the hollow organs like the digestive organs. Okay, so your stomach, your esophagus, your small intestine, large intestine has smooth muscle. Also your blood vessels, the arteries and veins have smooth muscle in the walls of them, which helps to move the blood along through the blood vessels. So cardiac and smooth muscle are not a big part of this chapter because you come back to that in other chapters with other systems. But skeletal muscle, we're going to go into a lot of detail with the skeletal muscle in this chapter. Okay, so we said that attached to the bones and skin of the face, voluntary. The organs of the muscular system are the skeletal muscles. So we have like the biceps brachii right here in your arm. That would be an organ. It's an organ because it's made up of more than one tissue type, right, working together for that specific function. So the other tissue types, we have nervous tissue, blood, which is a connective tissue, and other connective tissues that help to cover and connect the muscle to the bone and covering the muscle itself. Okay, so we'll go through the connective tissue coverings. We have the fascia on the outside of the muscle. It surrounds the muscle. It covers it. It protects it. Then we have the tendons. Tendons connect muscle to bone. Okay, so tendons are more like rope-like structures. For instance, here with the quadriceps tendon, which connects the patella to the quadriceps muscle, right? So it's kind of like a rope-like attachment. Aponeuroses are like tendons in that they connect muscle to bone, but they are flattened out like a sheet. Okay, so it's a flat sheet-like tendon. An example of an aponeurosis is here on the top of the skull, where the frontalis muscle, which is here on your forehead, the frontalis muscle is a flat muscle. It goes across the top of the scalp there, and it flattens out here, and then it connects again back here to the occipitalis muscle on the back of the head. So this aponeurosis is a flattened tendon that goes across the top of the head. Has the same function of a tendon in that it connects muscle to bone, but it's just flat. Okay, the coverings of the muscle. So some terms here that I want you to look at. Mysium. When you see mysium or myo, right, I want you to think muscle. Okay, so myo and mysium refers to muscle. We'll also go over another term that refers to muscle in a little bit. We'll look at sarco. Okay, sarco also refers to muscle. Right, so we have epimysium, perimysium, and endomysium. Okay, epi means above. This is the outer covering on the muscle. This is what separates the muscle from the rest of the body, that outer covering. The perimysium is inside of the muscle and it surrounds the fascicle. So this is a fascicle right here, which is a bundle of muscle cells. Okay, a fascicle is a bundle of muscle cells, and the perimysium surrounds that fascicle. Okay, then we have endomysium. Endomysium, so we have further bundling here, and on the inside of the fascicle is where we have the muscle cells. Muscle cells are also called muscle fibers. Okay, and so each muscle cell is then wrapped with the endomysium. So we've got epimysium, perimysium, and then endomysium. Okay, so those are the coverings for the muscle. Okay, the skeletal muscle is also called a muscle fiber. Okay, those terms can be used interchangeably. So muscle fiber and the muscle cell are the same thing. Okay, you probably remember from the histology lab that the skeletal muscle cells are multinucleated. What does that mean?
More than one.
Has more than one nucleus, right? So they have more than one nucleus. Here's a nucleus here. They're depicting one here. So along that muscle fiber, there will be more than one nucleus dispersed along. Okay, inside of the muscle fiber, you find it jam-packed with protein. There's lots of protein inside of the muscle fiber. And these proteins are in these long fibril arrangements. Okay, so inside of the muscle fiber is further bundling of the proteins in there. Okay, so notice that this is a fibril, myofibril. This is a myofibril. This is one, and what that is a bundle of protein inside of the muscle fiber or the muscle cell. Okay, the proteins in there are mainly actin and myosin. There's some other proteins as well, but those are the two main ones, are actin and myosin. Okay, actin filaments are the thin filaments. Okay, myosin filaments are the thick filaments. So we have actin and myosin, the thin actin, the thick myosin. Right? So here it's depicted with different colors. The myosin is this darker, like pinkish red color, and then the actin is this more like bluish color. So you can tell that those are thinner and these are thicker. All right. Also, so I mentioned the term sarco, meaning muscle. So we have sarcomeres, sarcoplasmic reticulum. Okay, so sarco is a term that we use especially for the muscles, and the sarcolemma, let's start with that. The sarcolemma, excuse me, is the same as the plasma membrane. So the sarcolemma is the plasma membrane of the muscle fiber. Okay, then we have the sarcoplasm. What do you think sarcoplasm refers to in relation to just a regular cell term? Right, the cytoplasm. So the sarcoplasm is the cytoplasm. Then we have sarcoplasmic reticulum. We had a similar thing. The endoplasmic reticulum, right? So we have sarcoplasmic reticulum, and is the same as the endoplasmic reticulum. Okay? And I think that's all of the sarco things there. Now, let's look at what this triad is here. So we have the T-tubules. So this is a T-tubule, which is a transverse tubule. It goes along the sarcoplasmic reticulum, or the SR. And then on both sides of the T-tubule is the cisterna of the sarcoplasmic reticulum. And so these three things together make up what's called the triad. So the T-tubule, and then the sarcoplasmic reticulum cisterna on both sides of the T-tubule. Okay? And that's important because we're going to talk about what happens with calcium ions in just a second, and how they use this to travel through to get down into where the muscle proteins are in here. Right, so you can see the thick and thin filaments there. Lots of mitochondria inside of the muscle fiber. Why do you think there will be so many mitochondria? What's their function? It's the powerhouse. It provides what? ATP energy, right? So you have to have plenty of mitochondria to provide enough energy for all of the muscle contractions that are going to be performed in this muscle cell, this muscle fiber. So lots of mitochondria. And the more that you condition yourself, the more mitochondria you will produce. So mitochondria actually self-replicate. They can make copies of themselves. And if you start exercising and you need more mitochondria, then they can reproduce themselves and make more mitochondria to provide you with that ATP needed for your activities. Okay, so looking here at the muscle fibers in here, and we're going to look at the sarcomere. So here's a sarcomere right here. And what the sarcomere is the basic contractile unit of the muscle fiber. Okay, so think about my fingers as a sarcomere. Okay, my thumbs, when my fingers slide past one another, my thumbs get closer together. That's what happens with the sarcomere. It runs from Z-line to Z-line, and when the muscle contracts, the Z-lines, which would be like my thumbs, get closer together. Okay? So the sarcomere is the basic contractile unit of the cell. That will definitely be on your test and on your exam. Okay? The sarcomere is the basic contractile unit of the cell. That's where shortening occurs. So the muscle is able to contract or shorten because those fibers slide past one another in the sarcomere. The sarcomere is made up of a pattern of light and dark bands. One way that you can remember this is that an I band is a light band, okay? So the I band is the light band. Light has I in it. Okay, so I, light band. The A band is a dark band. Dark has an A in it. Okay, so the dark bands are the A bands. And so it alternates light, dark, light, dark. Okay? In the middle of the dark band, the A band, is a line called the M line. That's the middle of the sarcomere. Okay, and that's where the filaments are held together in the middle. Okay, the Z lines are the ends of the sarcomere, and it kind of zigzags, so that's where it gets the Z line name from. The H zone is an area where you don't have overlapping of the thick and thin filaments. Okay, so the A band, the dark areas of the A band are where you have overlapping thick and thin filaments. But the H zone in the middle, there's no overlapping there. Okay, the light bands, the I bands, are only actin, right? The H zone is only myosin. The dark areas of the A band are actin and myosin, so thick and thin filaments. Okay, so Z line to Z line, I band, A band, I band. M line's right in the middle. H zone is the lighter area that's in the middle of the A band. Here you can see a slide of the sarcomere, and you can see the Z lines here, right? So those are the Z lines. This is the M line right in the middle. This is the H zone right here, right? This is a light band. This is a light band. This is a dark band. Okay, so I band, A band, and I band. Z line here. So the sarcomere goes from here to here. Okay, so the thick and thin filaments. So the myosin has these heads on it that form cross bridges or connections with the actin. So here's the actin here. Actin are these light blue proteins here. And actin is wrapped up with troponin and tropomyosin. So here's the troponin, these three protein balls here. It has three subunits. And this is the tropomyosin, is these long filamentous proteins. So that's a thin filament. This is a thick filament. The thick filament is made up of myosin only. Okay? And myosin has the heads there that are going to make those connections. So we've got thin filaments, myosin, thick filaments, myosin, thin filaments, actin, troponin, and tropomyosin. Okay? And they're all going to interact with one another for muscle contraction. All right, so when the muscle contracts, the filaments slide past one another in the sarcomere. Just like I said, my thumbs are the Z lines, right? And they're going to slide past one another like that. Okay? So how does that start? We need to go to the neuromuscular junction to see where that occurs. How does it get started? What's the impulse that stimulates the muscle to contract? Because we said this is conscious control, right? This is voluntary muscle. So when I think about contracting my muscle, an impulse is going from my brain down through my spinal cord and then out to my arm, and then it goes to my biceps brachii, and I'm thinking about it, and I'm going to contract my muscle. Okay? The impulse is coming here. This is the axon of a motor neuron. Okay, the motor neuron is carrying that impulse to the muscle. So this side here is the muscle fiber. The axon branches out into several terminals. So here we have a enlarged area of an axon terminal. Okay, the axon terminals, they remind me of like frog fingers. Have you ever seen like the tree frog fingers that have the little pads on them? So that's kind of how it looks. Okay, and so it branches out into those little pads, and each one is going to have this interaction with the muscle. The muscle side of the neuromuscular junction is called the motor end plate. Okay, so this is the motor end plate here. The axon terminal and the motor end plate do not actually touch each other. There's a little space there called the synaptic cleft. Okay, and they don't actually touch. There's a space. So in order for the motor neuron to interact with the muscle fiber, it has to send a neurotransmitter from the neuron to the muscle. And that neurotransmitter is a chemical messenger that goes from one cell to the other. So here, as the impulse travels down, we go up here and we have these synaptic vesicles here, which are going to move down to the bottom of the axon terminal. The vesicles are like little bubbles that are holding the acetylcholine, which is the neurotransmitter, and they release that neurotransmitter into the synaptic cleft. Okay, the acetylcholine diffuses across that space. So it diffuses. It goes from higher concentration to lower concentration across the synaptic cleft. And the acetylcholine binds to receptors on the motor end plate. Okay, receptors are proteins that stick out on the surface of the cell. And they have a specific shape. And that shape only fits one particular chemical. In this case, it is the acetylcholine, which is the neurotransmitter. Okay, so the receptor and the acetylcholine connect, and then that transfers the impulse from the neuron to the muscle. That's what stimulates the muscle to then respond to that impulse, is the acetylcholine. Okay, so it goes across, it binds to the receptors, and then once the acetylcholine binds to the receptors, that causes a change in the muscle fiber that is then going to lead to the contraction. Okay, so let's make sure we have everything here. We have the motor neuron, that's this part right here, the nerve that's going to the muscle. We have the motor end plate, which is the muscle side of the neuromuscular junction. We have the synaptic cleft, which is the space between the neuron and the muscle. We have the synaptic vesicles, which are the little bubbles there that are containing the acetylcholine, and it carries it and releases it to the synaptic cleft. And then we have the neurotransmitter, which in this case is acetylcholine. Okay, so acetylcholine is the neurotransmitter for motor neurons. Other neurons have other neurotransmitters, but for motor neurons, for muscles, it's acetylcholine. Okay? So we abbreviate acetylcholine ACH. Okay, so that's the neurotransmitter. All right, so the ACH binds to the receptors on the motor end plate. It causes changes in membrane permeability to sodium and potassium ions. Okay, Na plus is sodium, K plus potassium. Okay, so sodium and potassium ions. This generates the muscle impulse, which is also called the action potential. Okay, when we get to the nervous system, we're going to talk more about action potential. Okay, so for now, I want you to just know that it changes the membrane permeability and that impulse gets transferred over. The impulse causes the release of calcium ions. Okay, so the calcium ions are going to go from the sarcoplasmic reticulum down into where the myofibrils are to bind to the thin filaments. Okay, so calcium ions are going to go out of the sarcoplasmic reticulum and bind to the thin filaments. Okay, the excitation contraction coupling. So what we mean by that is the connection between the muscle fiber stimulation and the muscle contraction. So how does the muscle fiber being stimulated by the neuron lead to a contraction? Okay, so we mentioned the calcium ions in the sarcoplasmic reticulum, the SR. Okay, so they're going to be released from the sarcoplasmic reticulum, and the calcium ions are going to interact with troponin and tropomyosin to allow the contraction to occur. Calcium ions bind to troponin, so troponin is part of the thin filament. The calcium ion binds to troponin, and then that allows tropomyosin to be able to move out of the way and expose the myosin binding site on the thin filament. Okay, so look at this page in your book because I know the print, everything's too small for you to see from where you're sitting. So find this page in your book and let's go through this. So we're going to start out with relaxed muscle at the top. When the muscle is relaxed, okay, there's no connection there between the thick and thin filaments. Okay, the calcium ions are in the sarcoplasmic reticulum. Okay, then when the impulse from the neuron gets to the muscle and the neurotransmitter stimulates the muscle, then the sarcoplasmic reticulum releases the calcium ions, which go into the muscle fiber and bind to the troponin. So here, this blue protein here, it has 3 subunits. That's the troponin. The calcium ion is this lighter kind of bluish green. It binds to the troponin, and that causes the tropomyosin to move out of the way. And it exposes the myosin binding sites on the actin filament. The myosin binding sites are these little pink dots on there. All right, so this part is actin, that's actin, that's actin. These are the myosin binding sites. What that means, myosin binding site, is where the myosin is going to be able to connect to the actin. So the calcium ions bind, tropomyosin moves out of the way, and then the myosin is able to form cross bridges with the actin. Okay, so the cross bridges are when actin and myosin are connected. So let's say this part of my arm, this is the actin, the myosin is down here, and it attaches. That's the cross bridge. okay? Then it's going to have the power stroke motion. And what that is pulling of that actin. Okay, so once they connect, it's going to pull the actin and pull it back like that. That's that sliding action where we talked about in the sarcomere, the actin and myosin slide past one another and the Z lines get closer together. Okay, that's how it happens, is myosin grabs onto the actin and pulls it in that direction. So it pulls it over. And then notice here all along we have ADP plus a phosphate. Okay, so remember that ATP has 3 phosphates. How many phosphates does ADP have? ADP has 2. Okay, so ADP plus a phosphate. Okay, so that is where the phosphate has been removed from ATP, but it's still there. It's still in the general vicinity. Okay, so you have ADP plus the phosphate there. You still have ADP plus the phosphate. Still have it. Still have it. Then right here, this third phosphate gets added on to ADP to make ATP. And that allows the myosin cross bridges to, or the myosin heads to recock. So notice here that they're in this direction. The ATP attaches. It releases the phosphate again immediately, and that causes it to recock the head. So notice it's swung back in this direction. So it did power stroke in that direction, pulling on the actin. Then it needs to recock so that it can grab on again and pull again, right? To continue that shortening process. So the third phosphate is released from the ATP to make ADP, which allows the myosin to recock, and it starts over and it goes back through again. Okay, so this process could continue on until the muscle is fully shortened. Then relaxation occurs, right? And the myosin is no longer attached to the actin. This is called the sliding filament model of muscle contraction because the filaments slide past one another. So the muscle itself, it's not shrinking, right? It's not like actually physically shrinking into something less, right? It's just the fibers slide past one another and it brings the Z lines closer together. So remember, in the sarcomere, think about my hands being the sarcomere, and you have the fibers here, and my fingers are the Z lines, my thumbs are the Z lines, and those fibers slide past one another. That causes the Z lines to come closer together, and it allows the muscle to shorten, which is the contraction. So my biceps brachii here, when the fibers slide past one another, it shortens the biceps, and that's the contraction. Okay? Here you can see this is relaxed here, so you've got the sarcomere, the Z lines are far apart, and then when it starts to contract, they start sliding, and then fully contracted, they're all the way together. You can see here in the micrograph, you've got the Z lines here. When it contracts, they're sliding closer, and then they're fully contracted all the way together there. Okay, so that's the sarcomere with the Z lines coming closer together. Okay, this slide specifically talks about what's happening with the ATP and ADP, which we talked about. So you can see here, we go through this cycle of ADP to ATP to ADP around like that. Okay? So notice here that the linkage between actin and myosin cross bridges break, and then the ATP splits, and by it splitting there, it allows that head to it goes from this position to recocked like that, so that the next contraction can occur. Okay, then we have an enzyme, acetylcholine esterase. We know this is an enzyme. Why? What's something that gives it away? Right, ASE. Okay, that tells you that it's an enzyme. So acetylcholine esterase decomposes acetylcholine so that it can stop the contraction stimulation. Right? As long as acetylcholine is there binding to the receptors, then the contraction is going to continue. So it has to decompose the acetylcholine to be able to stop the contraction so the muscle can relax. So the neurotransmitter acetylcholine gets degraded. The calcium gets pumped back into the sarcoplasmic reticulum. The troponin and tropomyosin go back to covering the binding sites, and the muscle fiber relaxes. Okay, so it's a multi-step process to get back to relaxation. All right, so I'm going to stop here, and I'm going to write some stuff on the board. I want you to write this as well. And I'm going to draw the neuromuscular junction. So you need to draw this in your notes. And this is for a grade, so you're going to miss it. Okay, so here we have our neuron. Right, so this represents the neuron. Okay, this is the cell body of the neuron. There's the nucleus. This is the axon. It goes down and branches off into the axon terminals there. Okay, and the axon terminals are where we have that neuromuscular junction. So this is going to be the muscle here. Okay, so that's the muscle. And remember, we have a little gap there in between the muscle and the axon terminal. So this orange is going to represent that little gap. Does anybody remember the name of the gap? The space between the terminal and the muscle? Starts with an S. Synaptic cleft. Okay, the synaptic cleft is the space between the axon terminal and the muscle. All right, so we have our impulse, which is going along the axon, and it travels in one direction only. We'll talk more about that when we get to the nervous system. So the impulse is going in this direction. And when it gets here to the axon terminals, it causes the acetylcholine, which are in the vesicles, to be released into the synaptic cleft. So acetylcholine is here in vesicles. Right? And it gets released there and goes out into the synaptic cleft. Okay, the acetylcholine, we'll abbreviate that ACH. That is the neurotransmitter. Okay, the neurotransmitter is the ACH, acetylcholine. So it goes from the vesicles into the synaptic cleft where there are receptors specifically for acetylcholine in the synaptic cleft. And when the ACH attaches to it, it's then going to stimulate the muscle cell to continue on with that impulse, right? So that contraction can occur. All right, so let's make a list here of what's going on. So the impulse travels down the axon to the axon terminal. Okay, in the axon terminal is where you have the vesicles, the synaptic vesicles. Synaptic vesicles release acetylcholine into the synaptic cleft. Okay, number 3. Okay, so now the acetylcholine is in that space. It moves over by diffusion, and then the acetylcholine binds to acetylcholine receptors on the motor end plate, which is the outside of the muscle fiber. So that's the muscle fiber. Okay, so now the ACH has carried that impulse over from the neuron to the muscle. And remember that ACH is the neurotransmitter. Okay, then we have the sarcoplasmic reticulum. I'm going to abbreviate that SR. The sarcoplasmic reticulum is stimulated to release calcium ions, that's Ca+. So it releases calcium ions Right? And then the calcium ions are going to travel into the interior of the muscle fiber, where the myofibrils are, and attach to troponin. So calcium ions attach to troponin. Then the troponin causes tropomyosin to move, right? So the tropomyosin moves out of the way, and that exposes the myosin binding sites on the actin filaments. Can y'all see down here? If I write it down here, can you see that? Okay, so myosin binding sites are exposed on actin. Okay, the myosin binding sites are where the myosin heads connect to the actin, and it does that pulling motion, right, which causes the sarcomere to shorten. So we go back through that whole thing of the shortening of the sarcomere. Go over here. Number 8. Myosin forms cross bridges with actin. Number 9, sarcomere shortens. And that's contraction. When the sarcomere shortens and the filaments slide past one another, that's contraction. Okay, does anybody have any questions about any of the steps here? Pause this a minute.